Hello friends, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning platform. Today we learn about clonal selection theory. Lymphocyte specific for an antigen is activated upon encountering with antigen presenting cells loaded with an antigenic peptide. When the infection takes place inside the organism, the pathogen which enters inside the body will be engulfed or will be recognized by the antigen presenting cells, and it will be digested, and the part of the antigen will be processed and it will be displayed on the surface of the antigen presenting cells. And when these antigen presenting cells will be recognized by the lymphocyte, they undergo further proliferation, or they will be selected for clearing the infection at that moment. The concept of activation of lymphocyte is called clonal selection theory. The theory was put forth first by Niels Jern and explained further by F.M. Burnett in 1957. This clonal selection theory says that antigen-specific clones of lymphocytes exist even before the exposure of antigens, even before the infection by the antigens, and a large number of clones are generated during lymphocytic maturation to diversify the recognition of microbial antigens. According to this theory, the clone of lymphocyte with the specificity towards the lymphocyte is already generated in the body, and later they will be selected and will be expanded for clearing the infection. The specificity of each T lymphocyte and B lymphocyte is determined before its contact with the antigen by random gene rearrangements during maturation in the thymus for T lymphocyte or in bone marrow for B lymphocyte respectively. So this specificity is generated on the surface of the T or B lymphocyte by the gene rearrangement during the maturation of the lymphocytes. The role of antigen becomes critical when it interacts with and activates mature antigenically committed T or B lymphocytes, bringing about expansion of the population of the cells with the given antigenic specificity. So, the specific lymphocyte or the antigenically committed T lymphocyte will be selected, and they will be expanded, their population will be expanded, or they'll undergo repeated division with the same antigenic specificity for that antigen. In clone of lymphocyte cells, a group of cells or a lineage of cells clear the infection at that moment, at that instance, and acts as effector cells. These are also called as plasma cells. Another group of cells with the same antigenic specificity retain the memory of that pathogen for the further infection. Such cells are called memory cells. So these challenged T lymphocytes or B lymphocytes will be selected to clear off the infection. During that period, they increases their number with the same antigenic specificity, and in a population, one group will clear off the infection at that moment. They are called as plasma cells or the effector cells. The another group will retain that memory. It will remember that pathogen for the further infection. They are called memory cells. When the danger of infection is over, the extra plasma cells or the effector cells will undergo programmed cell death or apoptosis. The body itself, the immune system itself, will remove the extra plasma cells which are generated against the specific antigen after the infection is over, after the complete removal of the pathogen from the body. This is the diagrammatic representation how the cells they behave with the self-reactive lymphocytes, how they are removed from the body. At the same time, with the specific lymphocyte expressing the specific antigenic determinant, how it will be further will undergo proliferation to clear off the infection. The hematopoietic stem cells, they undergo hematopoiesis, or they undergo further maturation, and they give rise to myeloid primitive cell, and another one is the lymphoid primitive cell. So this lymphoid primitive cell further undergo the development, and they give the different type of lymphocytes. So these lymphocyte each will be having the specific receptor, or the specificity of each lymphocyte will be varying, and these lymphocyte, if they show the reactivity towards the self molecules, the body molecules, or the own tissue or the own protein, such type of lymphocytes will be removed from the body, will be removed from the body. Next, in a population of lymphocyte having the different types of specificity for different pathogen, they will be selected depending on the pathogen infection. For example, when the specific pathogen infection takes place, so the receptor recognizes the antigenic peptide, and when the specificity occurs, or these specific receptors containing lymphocyte will undergo further division, and they form the population of cell. This is called clonal expansion. All the cells will be similar to the same lymphocyte which has the specific receptor for the pathogen or the antigenic molecule, and they will be repeatedly divided, and they form a population of cell. So this is an example where they have taken the B lymphocyte. Here, by gene rearrangement, the B cells will take up the different receptors, or different specificity will, and these receptor they have, each cells will be having the specificity towards the particular pathogen. And when the pathogen 2 or the antigen 2 enters, here for example the B2 cells will be selected to clear off the antigen 2, and they undergo repeated division. And all the cells will be having the specificity towards the same antigen. And in this population of cell, some cells will be clearing the infection at that moment, and they are called as the effector cells or the plasma cells. They secrete, or they produces secretory antibody that flow in the body fluid, whereas the other group of B lymphocyte, they remember the pathogen for the secondary infection, and this group of the lymphocyte B lymphocytes are called as the memory cell. When the infection is completely removed, extra plasma cells, unused plasma cells which are generated against the infection of antigen 2 will be removed by the programmed cell death or by the apoptosis. So this is regarding clonal selection theory. I hope you understood this topic. Thank you.